Good afternoon to the entire audience. I am Patrick Martin. I'm an associate professor at North Carolina A&T State University in Greensboro, North Carolina. By training, I am a cancer biologist. I did my undergraduate work at Virginia Union University in Richmond, Virginia. Once completing my BS in biology, I attended the University of Virginia in Charlottesville, Virginia, and earned a PhD in cell and molecular biology. And at that point in time, I was the first African American to earn a PhD in cell and molecular biology from the University of Virginia. At that time, once I completed University of Virginia, I stayed at the University of Virginia and moved from the cell biology department over to a clinical department in pathology and began to study brain tumors, or glioblastoma multiforme. And that's where I picked up my cancer research experience. And since then, I've been doing cancer research at North Carolina A&T. Today, my talk will be entitled Examining the Chemotherapeutic Potential of Rosehip, rosicanina extracts, and triple-negative breast cancer. In my laboratory, we study aggressive tumors, and triple-negative breast cancer happens to be a very aggressive tumor or aggressive cancer. And we study this by using a lot of times we try to identify novel components or compounds, or in this case, bioactive compounds that can potentially serve as chemotherapeutic or therapy to known chemotherapeutic complexes. So breast cancer is the abnormal growth of breast tissues, and in the most recent cancer facts and figures information that's provided by the American Cancer Society in 2019, breast cancer was the number one site or the number one site of cancers in females. As you can see there in the top right, about 30% of all new cancers in females from 2016 to 2019 were in the breast tissue. Also, if we look at estimated cancer deaths, breast cancer is number 2 in estimated cancer deaths, and we're looking at the red highlighted material in the bottom right portion of the slide, in which we expect over 40,000, close to 41,000 cancer deaths each year, and that makes up about 14% of all cancer deaths. So as you can see, breast cancer is a major issue for female health, and it's definitely a major issue when it comes to cancers as they are related to females. Triple negative breast cancer, again, if you look, take a look at this in an ethnic or racial capacity, a lot of times we study cancers in our laboratory that have health disparities, and triple negative breast cancer is one of those cancers that has a health disparity. As you can see here, about 32% of breast cancers in African Americans are 32% of cancers in African will be breast death amongst African Americans. In this case, it's 19% of the deaths compared to 14% when you look at the entire population. So triple negative breast cancer it demonstrates a health disparity against African American females as well as Hispanic national females, as well as Native Americans and American. In addition to this health disparity, African American women have a lower 5-year survival rate, which is 81% compared to 92% among non-Hispanic white women. Why is there such a difference in survival rates? It's anticipated that it could be biologic or genetic differences, prevalence or risk factors, barriers to healthcare access, or later stage of breast cancer diagnosis. One of the reasons why we think there's a health disparity is because of the fact that triple negative breast cancer, which we will explain in more detail going forward, is you see it more likely in African American females than you do in other ethnicities. We can see here, if you look at just breast cancer, you have a non-triple negative subtype, and then you have a triple negative subtype, which is highlighted in the red box. The specific characteristics of triple negative breast cancer is that it lacks 3 known receptors for breast health. Those are the estrogen receptor, the progesterone receptor, and the HER2 receptor, or the human epidermal growth factor receptor. This makes triple negative breast cancer much more difficult to treat because close to 80% of your therapeutic options for breast cancer are targeted to either the estrogen receptor, the progesterone receptor, or the HER2 receptor. Triple negative breast cancer, again, is an aggressive form of breast cancer. It's highly metastatic, it's migratory, it's invasive, has a much poorer prognosis than other forms of breast cancer. There are very few treatment options, and you have a lower recovery rate. In African American women, we see triple negative breast cancer more in premenopausal women than we do in other age groups. And also, we see a higher prevalence in premenopausal women amongst African Americans than any other ethnic or racial group. And we have a higher rate of reoccurrence for triple negative breast cancer in African Americans, which is close to 40%, whereas in the normal population, non-African American population, it is 15%. So again, what makes triple negative breast cancer triple negative? So what you see is that your normal treatments, Herceptin and tamoxifen, are made against the estrogen receptor, the HER2 receptor, and as well as the progesterone receptor. But in this case, in triple negative breast cancer, as you can see here in the yellow cell on the right of the screen, it doesn't have any of these receptors. So if we try to use any of these treatments against it, they are not effective because the tamoxifen and Herceptin do not have a receptor to bind to, so they can be internalized into the cell and elicit their effect. So triple negative breast cancers are very challenging once diagnosed because they're very hard to treat. In addition, one of the major things that we see with triple negative breast cancer is one of its major characteristics, its ability to migrate and invade throughout the breast tissue and eventually external to the breast to other organs and organ systems. This makes triple negative breast cancer and any other cancer much more difficult to treat once it metastasizes and leaves its host tissue or its initial host tissue. And we see that approximately 15 to 30% of triple negative breast cancer patients actually develop brain metastases, which are very difficult to treat. And the life expectancy after metastasis is about three months to close to 24 months. So again, just typical breast cancer treatments. Your first line of defense is probably some type of surgery, whether it's mastectomy or some type of lumpectomy. You could also do targeted radiation. Chemotherapeutic, which is would be whole body chemotherapeutic. There are some hormone therapies that we could target for the estrogen receptor and the progesterone receptor. Therapies such as Herceptin, which can lead to targeting the HER2 receptor. However, all of these taken together are not that effective in triple negative breast cancer. So it lends to the fact that we need to find better ways to treat this very aggressive form of breast cancer. So this led to a new wing. It's not really a new arm. We've been doing it probably for about close to 10 years now in the department, where we look at natural therapeutics and their ability to potentially serve as a chemotherapeutic, chemopreventative, or adjunct therapy to known chemotherapeutic options. So why do we look at chemotherapeutics from natural products? One, they typically have lowerable unfavorable side effects than chemotherapy, and they typically have a low toxicity, and they typically have a much lower cost than some of our synthesized medicines. Natural therapies help provide antioxidant foods, which may have an improvement for cancer treatment. And additionally, a lot of your natural products have flavonoids and carotenoids, which have been demonstrated to be effective in preventing various forms of cancer, including breast cancer. So what we began to focus on serendipitously is Rosehip or Rosakinina extracts. And it just so happened I was on a committee when I first got to North Carolina A&T, and a student in the agricultural department was doing a master's degree looking at potential natural chemotherapeutics. And I was invited on the committee. And so I served on the committee, and I began to say, well, it is great that you have this empirical data, but we need to really begin to test it more succinctly and in known methods. In that time, we were looking at brain and liver cancer, but now we focused on triple negative breast cancer. So rosehips, which I typically call them, or Rosa canina, they're actually formed from what we call the Easter rose. It's a type of rose that buds around Easter time each year as we're entering into spring from winter. Have high levels of vitamin C and other vitamins, high levels of calcium, iron, and zinc, and as well as lycopene, which can be found in tomatoes and grapefruit, which has also shown anti-tumor properties. Common names for the rosehip are dog rose or wild rose. It's commonly used as a natural supplement to relieve pain, such as arthritic pain, menstrual pain, intestinal pain, colds, flu, sore throats. It's found in many teas. You actually find it in a lot of health and beauty aids, such as face lotions as well. Some previous work done in several laboratories have demonstrated that rosehip tea or extracts from rosehip can prevent breast cancer cell proliferation. And additionally, it can decrease abdominal visceral fat in pre-obese subjects. Previously, we've published the effect of these rosehip extracts on human brain tumor cell proliferation and apoptosis, and we've basically demonstrated that concentrations of these rosehip extracts can slow down and decrease the proliferation rate of malignant brain tumor cells. And we're going to do some of that work here in triple negative breast cancer. So overall, what are we trying to focus on in this study or in these studies that I'm going to show you today? We want to determine the potential of rosehip extracts as a regulator of cell proliferation, migration, and invasion of triple negative breast cancer cells. Today, we're really going to focus here on just objective number one, is to determine the effect of rosehip extracts on cell proliferation, migration, and invasion in breast cancer cells. We're also going to determine if rosehip extracts can serve as an adjuvant therapy with the commonly known breast cancer therapeutic agents to induce apoptosis. So we have these rosehip extracts, and we're going to expose them to human breast cancer cell lines. And we're going to focus mainly on, in the list there from top to bottom, HCC 1500s, which are a breast cancer cell line. They are technically not triple negative, but they are an aggressive form of luminal breast cancer. HCC 70, I will refer to these as 70s going forward. I'll refer to the 1500s as 1500s going forward. And then finally, HCC 1806s, a triple negative breast cancer. We also will talk about briefly the AG11132s. This is a normal breast tissue cell line that we always try to run in our studies when we're looking to this chemotherapeutic. So one of the first things we do, we'll do an MTT assay. We'll do an apoptosis cleavage assay to determine whether or not we're having an effect on cell proliferation and whether or not we're promoting apoptosis with rosehip extracts. So typically, most of our assays, cell-based assays, are done in a 96 well plate. You can see here that we will have our controls are serum starved. We have serum containing or plus serum. We have a known inhibitor of the mTOR PI3 kinase pathway, LY294002. We have a known inhibitor of the ERK pathway, U0126. And then we have our rosehip extracts, which are developed in, they're methanol-based extracts. And they are 1 milligram per mil, 250 microgram per mil, 25 microgram per mil, and 25 nanogram per mil. So the first thing we did an MTT assay, in which we just basically asked the question, do we see a decrease in mitochondrial activity, which we associate with a decrease in proliferation? If you look at the three figures here, you have the 1806s, the 70s, and then the 1500s. And if you notice on A and B in both of these, A is your minus serum control, then the B labeled bar is your plus serum control. This is basically our control with minus serum. We're serum starving the cells so they should not grow much. And then we'll give them a serum stimulation with the plus serum to ask the question, do the cells go ahead and grow? Then we begin to treat with, in order from higher concentration to lower concentration with your rosehip extracts. And what we notice here, if you look at all of the bars labeled C, is that our highest concentration of rosehip extracts is able to decrease the amount of cell proliferation in all of the cell lines tested. And again, we can have some decrease as well in lower concentrations, such as 250 microgram per mil, 25 microgram per mil, and 25 nanogram per mil. So this was pretty interesting for us to see that we actually can start with these rosehip extracts, expose them to the cells, and we expose them to the cells for 48 hours. And then we ask the question, do they slow or stop cell proliferation? And we can see here there's a dose-dependent effect that we can decrease cell proliferation. And at the highest concentration, we actually stop cell proliferation. So we were pretty excited about this. This was our first foray into the triple-negative breast cancer treatments with the rosehip extracts. So we know rosehip extracts can decrease cell proliferation, but the next thing you always think about, if these are chemotherapeutic, can they also promote apoptosis and kill these cancer cells? So the next assay we're going to look at, we're going to ask the question, can we actually kill or initiate apoptosis in these triple-negative breast cancer cells? So what you have here, again, you have at the top left, you have 1806 cell lines. Just next to that, you have HCC 70 cell lines, and at the bottom center, you have the 1500 cell lines. And what we're showing in this diagram, you have two lines. You have a line that has circles and a line that has triangles. The circle line is just looking at cell viability. So that's akin to our Mtt assay. Whereas the triangle line is looking at caspase-3-7 cleavage, which is an indicator of apoptosis in these cells. So we're looking at cell viability on the y-axis on the left, and on the right y-axis, we're looking at caspase cleavage. So when we get caspase cleavage, we know that we're entering apoptosis. Along the bottom, we're looking at our different treatment groups with plus serum on the far left. Then we move into 1 milligram per mil of rosehip, 250 micrograms per mil of rosehip, and then 25 micrograms per mil rosehip, and 25 nanograms per mil. And finally, on our far right, on each one of those graphs, we have 1 micromolar of staurosporine. This is it's a chemical compound that can elicit apoptosis in these cells. So it is what we call apoptosis positive control. So what we can see here in 1806 is that as you're going along, if you look at that top line there, when you treat with the plus serum and all the rosehip, the cells are pretty viable. So they're not dying when we treat them with rosehip. But if you look, and if you look at the line at the bottom of the graphs, you can see that that's measuring caspase cleavage. So again, when caspase is cleaved, in caspase cleavage in 1806 cells. When we treat with staurosporine, we actually can induce apoptosis in the cells, and you can see that the top line drops as far as cell viability close to 40 to 50%, and the amount of caspase cleavage goes up to close to 100%. If you look at HCC 70s and HCC 1500s, you see very similar trends in the line there, in which your rosehip extract is not killing the cells. It is not promoting death. It is only merely stopping the cell from growing. It's inhibiting cell proliferation, but not killing the cells. If you look at the Western blot over there to the right, we have HCC 1806s, then 70s, then 1500s. And basically, we're asking the question, are we cleaving PARP? PARP, along with caspase cleavage, are signs of apoptosis. And if you can look there at all the treated samples, only the column on the far right, which is actually treated with your staurosporine, you can actually see PARP being cleaved. And PARP uncleaved is 116 kilodaltons, and cleaved, it is 89 kilodaltons. So as you can see, that the take home from this is that, yes, rosehip extracts can stop these cells from growing, but they don't kill the cells. They're merely cytostatic and not cytotoxic. So rosehip extracts decrease cell proliferation in all the cell lines without inducing apoptosis. Rosehip extracts neither stimulate part cleavage nor activation of caspase-3-7 in human breast cancer cell lines. So what's next? We know we can stop these cells from growing, but we're not killing the cells. So we're happy that we can slow the cells and stop them from growing, but we're not killing them. So we next want to ask the question, what's the next biggest thing with breast cancer? It's the metastasis, the migration and invasion. Can we slow or stop these cells from migrating and invading? So the next thing we wanted to do is actually answer these questions. So initially, we'll look at migration. On the left here, you'll see little, it's a cell plate with 6 wells in each plate. And we have 2 little red lines. And basically what we do, we culture these cells with a plunger in the middle. And we eventually pull that plunger up, and that leaves a void. And we ask the question, can cells crawl into that void? And if so, we know they're migrating. And additionally, we did a hanging well, very similar to a trans well, except for this time we just ask, and the cells crawl in a 3-dimensional metrics more so than a 2-dimensional metrics. So the first thing we did, we looked at HCC 1806 cells. We cultured the cells regularly on a 6 well plate. The black space is the void. The green is the pseudo color we added to actually quantify the amount of cell migration. And if you can see here, we have 0 hour at the top and 24 hour at the bottom. And basically what we are looking at here, we're asking a question from 0 to 24, does that black space get smaller? You can see with minus serum, it doesn't much. Plus serum, it definitely does. And then we use our treatments. At 1 milligram and 250 micrograms per mil, we're able to decrease that migration significantly, almost stop it completely. With the lower concentrations, 25 micrograms per mil and 25 nanograms per mil, the cells still move and move almost as well as the controls. So again, rosehip at higher concentrations can at least block the migration of these cells. We next looked at HCC 70s, and we see a very similar event going on. Plus serum cells go ahead and grow fairly well. With the higher concentrations of rosehip, you're able to stop the cells from migrating. With the lower concentrations, you're getting some migration there, almost similar to what we see in our plus serum controls. The next thing we see is we're taking a look here at HCC 1500 cell lines. And again, very similar, plus serum, they go ahead and crawl together. And in the higher concentrations of your rosehip extracts, they don't crawl as well. So again, very similar. We're able to determine at this point in time that rosehip extracts can prevent migration. So the next thing we wanted to do is look at migration. Okay, so what we're looking at here, we did a hanging well assay, and we asked the question, can the cells crawl through a 3-dimensional matrix? It's a little paper membrane with holes in it, and can they crawl through and fall to the bottom? And if they can fall to the bottom, we know the cells are migrating. So that's exactly what we're asking for here. So what we're doing here, we took the same thing, 1806s, HCC 70s, and HCC 1500s, and you can see from left to right, you have no treatment, 1 milligram per mil rosehip, and 25 microgram per mil of rosehip. So we have a high concentration and a lower concentration. And what you can notice there is in that middle column, with the high concentration, you're able to stop the amount of cells that can migrate through in a 24-hour period. You can see the same thing. You can see some inhibition as well at 25 micrograms per mil of rosehip. And then those graphs on the side actually do a quantification of this. And we can see that we have significant inhibition of migration in all 3 cell lines tested. And we actually have that with the higher concentration and the lower concentrations of Rosehip. So at this point in time, we're pretty happy. We can stop the cells from growing. We can stop the cells from migrating. So the next thing we want to ask the question is, can we stop them from invading? So we use the same transwell or hanging well assay. We had that paper lining again with the holes in it. This time we coated the top part of that with the extracellular matrix membrane, and we asked the question, can these cells degrade that extracellular matrix membrane, crawl through the holes, and fall to the bottom? If this is so, we know the cells have the ability to metastasize. They can actually leave the breast tissue, get into the bloodstream or lymphatic vessels, and move throughout the body. And this will mimic, this is an in vitro imitation of metastasis. So we did the same thing, very similar. We looked at rosehip extracts. Again, you have HCC 1806, HCC 70s, and 1500s from top to bottom. 3 columns once again, no treatment, 1 milligram per mil and 25 microgram per mil. And this time we let the cells go for 48 hours because we need to have them time to see if they could actually degrade the extracellular matrix. And again, what we noticed is that although it's not as apparent as the migration, but the higher concentration of 1 mg per mil can actually slow down the ability of these cells to migrate and invade. So we know we can stop the cells with Rosehip from growing. We know we can stop their migration, and we can slow their invasion down as well. So it has potential to be a fairly good chemotherapeutic agent. So we looked at our objective 1. We're demonstrating that Rosehip can block growth, can block migration, and can slow invasive migration in the breast cancer cell. So what's next? Well, the one thing we want to do is ask a question. We understand people are hesitant about taking natural products, but can we use the natural products in combination with a known chemotherapeutic agent to see if you can have synergistic effects and better the effects of both the rosehip extracts as well as the known chemotherapeutic agent? So we actually use doxorubicin. You can see it here. Basically, it's known. It's a drug that's commonly used to treat breast cancer, and it commonly treats triple-negative breast cancer as well. It has a high amount of toxicity to non-cancerous cells as well. And previous studies have shown that quercetin, another natural product, along with doxorubicin, can actually have synergistic effects in reducing breast cancer cell growth and also decreasing some of the toxicity of doxorubicin. So what we want to do is ask a question: if we combine rosehip and doxorubicin together, can we decrease cell proliferation even further? And can we also begin to promote PARP cleavage, which is a sign of apoptosis? So the graphs at the top, again, are 1806, 70s, and 1500s from left to right. And the one thing I really want you to focus on is the far right column on each graph. That's where we use rosehip and doxorubicin together. And in all those cases, it has a synergistic effect to reduce the amount of proliferation even further. Alone, you can see the 1 mg per mil rosehip and the 20 micromolar doxorubicin, which are the third and fourth columns in each graph, respectively. They can reduce cell proliferation to below baseline levels, but together, when you combine them in the final column, they actually reduce proliferation even further. And if you can look at the Western blots, what we can also see is that they are actually begin to, at 24 hours and somewhat at 8 hours, the combination of rosehip and doxorubicin begin to show PARP cleavage. So together, they're synergizing to not only inhibit cell proliferation, but also begin to promote apoptosis, which rosehip extracts were not able to do on their own. We also decided to look at these in combination in migration experiments. Again, we have 1806 and 70 cells, 0 hours at the top, 24 hours at the bottom. And again, if you look at the far right column, you can see that together, rosehip and doxorubicin are able to decrease cell migration greater than they are as individual agents. And we've quantified that in the graphs to the right. And we can see the same data with the 1500 cells. We actually see that Rosehip and doxorubicin alone work well in inhibiting migration in these cells, and together you don't see a huge synergistic effect. So together, our conclusions are that Rosehip extracts decrease cell proliferation, migration, and invasion of these triple-negative breast cancer cell lines. They do not promote apoptosis in breast cancer cell lines. However, together, Rosehip extracts synergize with doxorubicin to inhibit cell proliferation and begin to promote apoptosis. And doxorubicin in the combination with Rosehip extracts prevent cell migration in breast cancer cell lines. So finally, with the little time I have left, we always like to ask this question, and every time we try to publish something, reviewers always ask, well, what's the effect of these Rosehip extracts on normal breast cells? Are you killing those as well? Are Rosehip extracts, although they're a natural product, are they going to have the same cytotoxic effects on normal non-cancerous cells? So we are fortunate enough that we have some breast stem cells, and they're really just breast cells, and we can run these sorts of assays. So again, right here, we ran a cell proliferation assay. In the middle there, we're looking at PARP cleavage and these are the AG11132 cell line, which is our normal breast cell line. And we just wanted to ask a question: do Rosehip extracts prevent the proliferation of these normal non-cancer cell lines, and do they also begin to promote apoptosis? So in the left graph there, you see that higher concentrations of Rosehip extract can decrease cell proliferation in this normal breast cell line. However, if we look at the Western blot there, the middle Western blot, which looks at PARP cleavage, we only see PARP cleavage in the far right column, suggesting that Rosehip extracts, just as they did in the cancer cells, do not promote apoptosis in the normal breast cells. So we're pretty happy to see that they can slow the growth of the normal breast cells, similar to what they do in cancer cells. However, they don't promote apoptosis, which is good for cytotoxic side effects. So just to try to wrap up in the overall conclusions, Rosehip extracts inhibit cell proliferation, migration, and invasion without promoting apoptosis. These extracts synergize with doxorubicin to inhibit cell proliferation, promote apoptosis, and prevent cell migration. Rosehip extracts do not kill normal breast cells. They merely delay their proliferation. And in conclusion, I'd just like to acknowledge most of the people who did this work. Patrice Cagle, she did close to 80 to 90% of this work when she was a PhD student in my laboratory. She is now a postdoc / associate at North Carolina Central University. Patrice did exceedingly well. She published 3 papers. She was able to win an NSF Graduate Research Fellowship Award before she moved on to her postdoc. Adamoli Shofalui, he did some of the initial triple-negative breast cancer cell migration experiments. He is now finishing up his residency at Emory University as an orthopedic surgeon and is the chief orthopedic surgery resident at Emory currently, and has already got a fellowship for ankle and foot surgery. Others, we've collaborated to help us look at some of the bioactive components of Rosehip, as well as we like to thank Dr. Keith Schimmel, who at that point in time was the director of the PhD program, and Dr. Saran, who at that point in time was dean of the graduate college. They really helped us in seeking our funding and making sure Patrice had the funding she needed to be successful. This work was supported by a Title III program. It was supported by the National Science Foundation Graduate Research Fellowship Program to Patrice, as well as the NIH Bridges-to-Doctorate program that I serve as the PI of, and an NSF Alliance for Graduate Education program that I serve as the PI of. And I would like to thank everybody and open the floor for questions right